Hello, everyone. My name is Callum, and my project here at SAMS is understanding and exploiting stoichiogenomics to harness reactive nitrogen in sustainable food production systems. I'm a slightly different flavor of the month to a lot of people here. I don't work with algae and I don't work with seaweed. I work with polychaete worms, which has led to a lot of people here affectionately dubbing us as Team Poo or Ooh, you smell gal in my lab. So just a bit of a background on kind of what we do here. So we looked a couple of years ago and we saw that there was a trend kind of occurring as fish farming kind of moved more towards the coast and then eventually onto land. And that is, what do we do with the waste that is currently being produced? So research has already built kind of the past 10 to 15 years on deposit feeders being good candidates for bioremediation systems. So consuming the waste, getting rid of it, and letting it being somebody else's problem. There was recent research done in the tropics on sea cucumbers that are really effective at doing this. And so we thought, well, why don't we, take this to the west coast of Scotland, decide, one of the biggest fish farming places in the world at the moment, and create a kind of temperate water system for this. So studies have shown that polychaetes can be effectively reared to a healthy size on fish waste. And in, I think it's Denmark, a company called Marina farm Arnica marina, which is just sandworm, and they cultivate them for their blood oxygen product. They are exceedingly high in hemoglobin levels. So these are then sold on to pharmaceutical companies and are often used to deliver, things like blood transfusions or that sort of stuff, just purely because of the high content. So what we see up the top right here is ProCate semi-moist feed, and it may be familiar to a few of you, but it's a shrimp maturation diet. So there is a compound in polychaetes that allow that facilitate the maturation of shrimp, and that's how you would push them on to harvest size and harvest weight. So there's a demand here for polychaete-based feed products. So what we want to do is create a system that bioremediates the waste from your fish farm. You can then harness the polychaetes from that and then refeed them to your fish. So it's circular economy principles. This is kind of the general overview of the PhD itself. So the hypothesis is that deposit feeding invertebrates can utilize and exploit the enriched GC content of sediment bacteria impacted by high nitrogen availability, so things like fish waste, and transcribe this increased availability into their genome and proteome to overcome the limitations. So the general thought is if you're feeding worms, you're feeding the worms, but what we want to change that is from feeding worms to supplementing the bacterial communities that we see in the sediment and the gut, because they facilitate the transcription and the uptake of nitrogen and these carbon products from the waste and into the polychaete wall, or body wall, sorry. So now that we've decided this is what we're going to do, we have all the worms in the world to choose from. What do we choose from? So research has been done extensively into these three, not exclusively, sorry, because there is loads all over the world. So there's Hedestia diversicolor, or mudworm, or harbor ragworm, if you want to call it. You would usually use it for fishing. It is very, very common around coastal waters here and reaches maturity very, very quickly. And they deposit feed and filter feed. They display unique filter feeding behavior that allows them to take particulates from the water columns. That makes an excellent, an excellent candidate for what we want to use it for. There was also Olitavirans, which a lot of people have used recently, but they are very aggressive little things and they will bite. So I don't like using them. And they tend to kill each other before you get any meaningful data out of them. And then obviously Arnica marina, which we spoke about earlier, but they burrow head first and feed on particular organic matter and sediments. So if you're feeding from top down, you want something that will take it before it penetrates the sediment and makes the sediment potentially toxic. Obviously, it really depends on what you're looking for in your system, but these are the three that we were looking at here. So just before I give a little outcome, we've run 2 experiments so far, with some data going to come out of them kind of in the next month or 2, I hope. So experiment 1, we fed 4 different aquaculture waste diets from Scottish waters. So they were salmon feed, they were salmon waste, lumpfish waste, and shrimp waste. So the shrimp waste came from our circulating aquaculture facility out in St Andrews. The lumpfish waste came from Otterferry Seafish down in Inverary. and the salmon waste came from Scottish Sea Farms at their Big Barcaldine hatchery less than 10 miles up the road. What we saw in this was that we could effectively rear these polychaetes on all of these different waste types. Didn't matter what it was, they all at least doubled in growth, which was really, really nice to see. But the difficulty we had there was survivability. Some of them had maybe a 30% survivability, and I think that was down to being a bit heavy-handed with my feeding. I think it's safe to say we were feeding them once a day, and basically just nutrient bombing them. And if you, as algal science, if you nutrient bomb something, you tend to kill it. So it's no different from worms. From experiment 2, we took that and went, what we'll try is try different dosing regimes and see what kind of sticks. So we fed, the same amount of biomass, but over a longer period of time to supplement the bacterial communities in the foregut, the sediment, and all the things we basically want to see. And this experiment actually finishes tomorrow. So yeah, once there's a bit more data out there, it'll be nice to be able to publish that and for you all to look at it. In addition to this, we're also waiting on redox readings coming back and CNN data to discover whether the worms actually bioremediated what we saw. The water chemistry points to the fact that they did, but we need to check the sediments as well to see whether there's been, an upwell of H2S in the sediment and whether it's been, oxidized and transformed into something else. So by the end of the month, this will be tied together quite nicely. And this is where CCAP-ARIES comes in. So before we started, I wasn't really sure, sure, we can feed these worms, but we need to be able to make sure that they'll actually, be palatable to fish and provide something to the farmers. Fish farmers are hard sell half the time. So being able to prove to them that what we say will happen. So as Matt showed earlier, I'm using GCMS. We're basically going to profile the lipids and the proteins out of it. And because this is slightly different, this is totally new for SAMS. Dr. Davies already, worked on a method for black soldier fly larvae. So we're looking at doing, experimenting with this one and another one led by Dr. Ross, who is basically specifically, changed a method for us to help us, derive the data that we want to see. We're also looking for amino acid analysis, and it's something that we are still talking to CCAP-ARIES about, where they want to expand out and fully commit to something like this as well. So there could be something, in here in the future, but it's really handy being able to just go up the stairs, and everything's in one place that we need. So once that data comes out as well, that'll be really, really nice to see. So the future steps from what we're doing just now is, this is kind of the proof of concept, and what we're going to go do later is we're going to go into a microbiome study of everything that we've done to try and unlock the mystery behind deposit feeding nutrition. So the entire GC content theory that I was talking about earlier, and the sediment gut interactions in the microbiome, and basically prove that, it's the bacteria that are key to deposit feeding nutrition. We also want to do stable isotope analysis to have a look at tissue turnover time, and see how quickly we can make a harvest cycle happen, and just try and make sure that we're constantly producing. And then finally, as a bit of a pet project, I'm going to create a database to analyze the GC content and its relationship with nitrogen, and this will hopefully strengthen the case that we're putting forward, which will hopefully flip deposit feeding nutrition on its head completely. So it's a really exciting kind of time to be ahead of the curve on this, and we've had quite a few companies come in and say, come back to me in 5 years, we're quite interested in what you have to say. So yeah, absolutely, we're enjoying working on it, but there's a whole lot going on out with what I've presented today, which is exciting. So I just want to thank my supervisors, Dr. Georgina Robinson and Dr. Adam Hughes, and obviously CCAP as well, who have allowed me to speak today. And I want to give a special mention to RASTEC as well for being a really big funding partner in my research so far and enabling me to, do everything I've done so far. So I'll take a quick couple of questions that you may have about my work as well. Nothing to talk about.
The naive audience in terms of worms.
Yeah, it's a tough subject, especially considering the different strands that we've got. Microbiome, we've got genomics as well coming in there. We're going to have proteomics. There's a lot of different threads going together, so it is quite a wide ranging project. Yeah.
Or maybe, I don't know if you want to talk a little bit about fuel cells that you're also playing with. We haven't.
Yeah, I won't say too much because it's still in its infancy, but we do have another 20 tanks set up. We are prototyping microbial fuel cells. So what we're basically doing is we're trying to harness electricity from the microbes in the sediment, which will also feed into this system where you will be able to create an entire prototype that will remediate your waste, be able to produce worms that you feed to your fish, and power itself, all in the power of polychaete worms and poo, which, you know, is crazy. It's absolutely crazy, right? You know, so yeah, we'll have some data coming out of that, but yeah, it's a really exciting time to be part of, you know, the stuff happening in the CCAP areas to enable us to do this.
No, thank you very much.
Thank you.